Hello, everyone. Good morning, good afternoon, good evening. This is Dr. Kaz, and we are going to continue on with Chapter 24, our urinary system. And we're talking about substances with regulated reabsorption. Just to review, reabsorption is the process of substances moving from the tubular fluid back into the blood. So that is reabsorption. So let's talk about sodium reabsorption. Sodium is an important ion, as you know, the importance of it in regards to both neurological function, the proper physiological functioning of our neurons when we're trying to generate action potentials, but also it does play a role too, all right, when we're trying to use our muscles because we need an action potential to stimulate the muscle to contract. So sodium reabsorption, anywhere from 98 to 100% of the sodium that was filtered into the tubular fluid will be resorbed from the tubular fluid back into our blood. So we're going to see, all right, along the entirety of the nephron tubule, all right, but mostly, a lot of things mostly happen in the proximal convoluted tubule. But most of this reabsorption occurs there. So the nice thing about this is we're going to deal with a concentration gradient once more. So inside the tubule cell, the cells that line our nephron tubule, all right, the sodium concentration will be low compared to outside, in which we'll see a high sodium concentration in the lumen and in our interstitial fluid. So of course, once we are able to get sodium to move across the plasma membrane of the cell, right, it's going to go from an area of high concentration from the lumen there, from the tubule fluid into the cell, an area of low concentration. So it's going to move down its concentration gradient. And so it does so through facilitated transport. Okay, it's still a passive process, but when we talk about facilitated transport, what we mean is, all right, the movement of whatever substance we're talking to needs help. And our facilitated transports are going to involve transport proteins. So in the basolateral membrane of these tubule cells, we have, we recognize these, right, our sodium potassium ion pumps. And so they're going to move our sodium from an area of high concentration to an area of low concentration. And we know that 1/3 of a neuron's energy expenditure is used to operate these pumps. And also we'll see a substantial amount of energy being utilized to operate these pumps in our kidneys. Now, when we get towards the end, right, of our nephron tubule here, we're going to see, the role of hormones and how they're going to regulate sodium reabsorption. So aldosterone and our atrial natriuretic peptide, that is that hormone that is generated by the atria of the heart when we have an increase in blood pressure. Aldosterone we'll see aldosterone's presence when our blood pressure has reduced or our blood volume has reduced. So of course, obviously the dietary intake of sodium plays an important role too. Folks that have a high sodium diet are discouraged, especially to have a high sodium diet if they have a high blood pressure. Because we'll talk about this, how our follower, water, is going to go where the sodium goes. So our two hormones, aldosterone and our ANP, right, aldosterone, we saw in chapter 17, is produced in the adrenal cortex. It's a mineralocorticoid there. And so what it does is it's going to make more of those sodium channels in the luminal plasma membrane. And that's going to help, that's what I was talking about before with the facilitated transport of sodium from the tubular fluid in the lumen into the cell. And then also it'll help to make sodium potassium ion pumps. And so where do we find, all right, these pumps in channels that we speak of for aldosterone? Well, in our principal cells. And they'll be at the distal end there of our nephron tubule construct. And so their job is to increase sodium resorption. And of course, if we're increasing sodium resorption, we're taking sodium out of the tubular fluid, putting it into our blood. And therefore, guess what? Here comes the follower. Water is going to follow sodium from the tubule lumen into the blood, and therefore it'll help to increase blood volume. Therefore, it'll help to increase blood pressure. We get to talk about more of this in much more detail in later chapter. All right, and of course, when we talk about the movement of water, we have to talk about the passive process of osmosis. That's the movement of water from an area of high concentration of water to an area of low concentration, right, across a semi-permeable membrane. And you could also say, when we're discussing the movement of water, that water is always going to want to move to the area of a higher solute concentration. In this case, our solute will be our sodium. And so water follows the sodium. So AMP, the atrial natriuretic peptide, right, this one, okay, is going to be seen being produced, like I said before, when our blood pressure has increased, our blood volume has increased, we get an increase in stretch in the atrial walls of our heart. So this hormone is going to be released, and it is going to inhibit the resorption of sodium in our proximal convoluted tubule, in the beginning of our nephron tubule, and then at the very end where the collecting tubules are. AMP will also inhibit the release of aldosterone. So it seems like it's the no fun hormone, likes to inhibit a lot of things. So if it's inhibiting the release of aldosterone and it's preventing the resorption of sodium, that means sodium stays in the tubular fluid, and then sodium will be excreted in the urine. Well, guess what? Where sodium goes, water follows. And so water will also be excreted in the urine. So as a result, and we've talked about this before, how AMP will increase our glomerular filtration rate. So here's a quick picture of our sodium resorption. Keep in mind, the majority of sodium will be resorbed here in the proximal convoluted tubule, right? About 65%, 25% in the loop of Henle or our nephron loop there. And then depending on what is going on in our body, right, whether we are hydrated or dehydrated or whatnot, will determine what amount of sodium is resorbed and what amount will be excreted. And so that is going to include our distal convoluted tubule, our collecting tubule, and the collecting duct. So here's an awesome, cool picture. I'll zoom in here a little bit. And we'll start off here with the tubule fluid. And so here's our tubule fluid loaded with sodium. We just filtered it out of the blood. Sodium is a very small, all right, solute. So it can make it through our filtering system, or filtering membrane, excuse me. So now we have lots of sodium here in the lumen of our proximal convoluted tubule. So of course, sodium will move across the luminal membrane here of our tubule cell, and it's going to need help from our transport protein here for facilitated diffusion. And it moves into the cell where the cell has a low concentration of sodium. Well, to get it out of the cell into the interstitial fluid, we rely on the sodium potassium ion pump. That is going to be our active transport. So it is going to move up against its concentration gradient. So while sodium is being pumped out, we're going to pump in potassium. We've seen this before. So potassium, as we know, banana floating in the ocean, right? Our potassium has a high concentration inside the cell. So then sodium will then diffuse into our blood and resume its path along, all right, the peritubular capillaries back into our bloodstream. So we're going to see about 65% of the sodium will be resorbed in the blood, and that's just in the proximal convoluted tubule. Now, when we get to the end here, all right, near the distal convoluted tubule, our collecting tubules and ducts, right, we're going to see, depending, like I said before, what the body's need is for sodium will determine what's going to happen. So in this scenario here, what we're seeing is sodium moving from the tubular fluid into our bloodstream. So again, here's our sodium traveling through in our tubular fluid, high concentration of sodium, right? Because a lot of times when that sodium has left the proximal convoluted tubule, water follows and it goes with it. And so that means the tubular fluid becomes more concentrated. So what we're going to see here, we have a high number, concentration of sodium, again, out here in the tubular fluid. Then we have our nice, all right, transport proteins here. Sodium then enters into our principal cells here. And then, of course, the principal cells have a low concentration of sodium, so we move down our concentration gradient. Same scenario that we saw before. Sodium will move across the basolateral membrane here, and then it goes into the interstitial fluid, and from there, it goes back into the blood. Now, this picture here is showing us the role of our aquaporins here and how they allow the movement of water across the plasma membrane. And you can see right here, all right, water is going to move across the plasma membrane, the semi-permeable membrane, according to the definition of osmosis, and it's going to go from an area of high water concentration to an area of low water concentration. So if sodium is going this way into our blood, water wants to follow. So how can we make this process easier? Okay, well, no problem. We can have aldosterone help us out. Okay, so aldosterone, our mineralocorticoid hormone that's produced in the adrenal cortex, we're going to see the role that it plays here in the principal cell. It's going to insert more transport proteins into the plasma membrane. So we'll see more channels here for sodium, and then more pumps here along the basolateral membrane. And then also at the same time, all right, this will allow, okay, more sodium to move across the plasma membrane of the cell, allowing for water to move along with it. Okay, so here's a nice slide to kind of show you what happens when we're dealing with water. Remember, water follows, water follows. So we saw 65%, right, reabsorption of sodium in the proximal convoluted tubule. So we'll see roughly the same number here, about 65%, right, as sodium is moving across the proximal convoluted tubule back in the blood, water will also follow it. Now we see a lower number here. We'll talk about this a little bit later, okay, for water reabsorption in the nephron loop. We'll get into that in a little bit. But again, right, depending on what's going on in the body will determine, all right, what we're going to see with water's reabsorption in our convoluted tubule, in the collecting tubule and in the collecting duct. But you'll notice here, all right, this slide is showing us what can influence water's movement when we get to the collecting tubule and the collecting duct, all right, antidiuretic hormone, ADH. This hormone is going to increase the number of aquaporins. Aquaporins are those channels designed, all right, to help facilitate the movement of water across the plasma membrane. So the more aquaporins that are available in the plasma membrane allows for water to easily move across the plasma membrane. So ADH helps to increase that number. So by increasing the number of aquaporins, we can increase water's reabsorption into our peritubular capillaries there. All right, what about sodium? Well, we just talked about sodium. What about potassium? All right, potassium is interesting because it is going to be reabsorbed and secreted. Okay, so and basically whatever's going on with sodium is going to play a role in potassium's movement. Right. So when we're looking at potassium here, you can see this number here, about 60 to 80% is going to be resorbed in the tubular fluid. So sodium, as you know, when it crosses over, all right, into the cell, water follows. And like I said before, as water follows, whatever's left behind becomes more concentrated. And so that more concentrated tubular fluid is going to alter now, all right, the concentration gradient. And so obviously, if we start to change the concentration gradient, then we're going to influence the movement of other solutes, other substances that are in that tubular fluid. So of course, all right, when we're dealing with potassium, potassium is going to move down its concentration gradient. So in this case, though, okay, potassium is going to move, all right, from the tubular fluid towards the interstitial fluid, but it's going to take a route of the paracellular direction. And what that means is it's going to go in between the cells. So we saw that there's two ways that substances can move from the lumen, all right, of the nephron into the bloodstream. It can either go the paracellular route, which is in between the cells, or the transcellular route, which we've just been talking about the majority here of, where it just moves across the plasma membranes of the cell. And so in this case, we'll see potassium moving the paracellular route. Now, when we get into the nephron loop, the loop of Henle, all right, specifically the thick segment, right, we will see another 10 to 20% resorption going on. But when we get to the collecting tubule now, all right, we're going to see whether secretion is going to occur or resorption is going to occur. So we're going to be dealing with the intercalated cells. And so these cells are constantly going to be resorbing potassium. If I said calcium earlier, I apologize. I don't know why I'm on a calcium kick, right? But potassium. So the intercalated cells are resorbing potassium all the time, right? And as you know, potassium levels are much more tightly regulated, and more physicians, more medical personnel are more concerned about potassium levels necessarily when they're more so than they are sodium levels. Not to say that they're not worried about sodium levels, but it's the potassium levels where many healthcare professionals are concerned about. Perfect example is lisinopril, which is a blood pressure medication, but one of the side effects of taking that medication is, all right, it can mess around with your potassium levels. And when you mess around with potassium levels, then you mess around with the proper functioning of certain effector organs, namely muscle, specifically cardiac muscle. If you already had high blood pressure, then you don't want to be messing around with the heart. So that's one of the things you have to take into consideration. So our principal cells now, all right, they're going to be secreting, all right, potassium. But that depends. It changes. And a lot of it is going to depend, again, on the body's needs, what other hormones are going to be being secreted. For example, aldosterone. Aldosterone is going to be highly influenced by elevated levels of potassium. In fact, that is the number one, all right, stimuli for aldosterone secretion. So if your potassium levels are elevated significantly, right, then that's going to trigger aldosterone to be secreted. And we're going to talk about its effect here in a moment, all right, but we'll see, okay, obviously if our potassium levels are elevated and high, then we're going to want to get rid of potassium. Think of your urine as like a dump site. If your body has too much of stuff, because we talked about earlier how the kidneys basically but that's their job, is they're a filter. And so it's going to filter your blood, right? So there's too much of something in your blood. If the kidneys can, we'll try to offload it and dump it into the kidneys and excrete it out into the urine. And so that's what we're going to see here. We're going to talk about this in a moment. But the cells that are going to be secreting potassium here, right, are going to be our principal cells. The cells that are going to be resorbing potassium are the intercalated cells. All right, so here we can see potassium movement, right? 60 to 80% resorption in the proximal convoluted tubule, 10 to 20% in the nephron loop, and then depending on what's going on, all right, in the distal part, of our nephron system, the collecting ducts and the collecting tubules here, right, we're going to see what's going to happen. We're dealing with our type A intercalated cells. They're going to be resorbing potassium all the time. And then depending on what's going on with aldosterone, will determine how much, all right, potassium is going to be secreted by the principal cells. All right, a couple other things. How about calcium and phosphate? Keep in mind, right? Our kidneys are the filtering system. Our urine is the dump site if we have too much of whatever. So during the filtration process of our blood, about 60% of calcium and phosphate are going to be dumped into the tubular fluid there. Right. A lot of calcium is, well, I shouldn't say a lot of calcium, right? But a significant amount of calcium is just free floating in the blood plasma, okay? But we also have calcium binding proteins that are in our circulation. If you recall, calsequestrin and calmodulin are two calcium binding proteins. We saw both of those in the sarcoplasmic reticulum, terminal cisterna, in skeletal muscle cells. We saw calmodulin in the sarcoplasm of our smooth muscle cells. Right? But point being is, when you go and get your blood drawn and they look at different levels of certain ions and they see, oh, your calcium is at this number here, that's the unbound calcium. That's just the free-floating calcium. Okay, so 60% of calcium and phosphate goes into the filtrate. And then what we'll see, all right, a significant amount, all right, because calcium, again, is a small molecule, right, will pass from the glomerular capillaries into our tubular system. So this brings in, all right, but way back in chapter 7, all right, we talked about what happens, what hormones are released when you have low blood calcium levels. Calcium is important, folks. We need calcium for, all right, one of the ingredients for bone, okay? We also need calcium for the proper functioning of our neurons. Hello, synaptic knobs, what's going on there? We also need calcium to help remove, all right, the troponin, tropomyosin regulatory proteins off of the myosin binding sites on actin. We need it for blood clotting, okay? And we need it for, I'm forgetting my fourth one. It'll come back to me. Anyways, so our parathyroid hormone is going to regulate, all right, the amount of excretion for calcium and phosphate. So parathyroid hormone, also known as PTH, is going to inhibit phosphate resorption in the proximal convoluted tubule. So if we're inhibiting the resorption of phosphate, right, in the proximal convoluted tubule, it'll stay in the tubular fluid. More on that in just 1 second. But what we'll see is, all right, PTH is going to increase the resorption of calcium, right, in the distal convoluted tubule. So what we'll see is we're going to keep the calcium, we're dumping the phosphate, and that's important because if the phosphate was present, then, all right, calcium could combine with the phosphate to help form those hydroxyapatite crystals, right? And that would be deposited in the bone. But we don't want calcium being deposited into our bone because don't forget, our blood calcium levels are low. We are hypocalcemic, low blood calcium levels. So we need calcium in circulation. And so phosphate's hanging out and is running around. It'll grab the calcium and come on, let's go. We're going to go to the bone and we're going to start making bone. No, we don't need that. So we dump the phosphate, we keep the calcium. So we decrease, all right, calcium deposition in our bone tissue. And where else can it go if it can't be deposited in the bone tissue? It's going to float in the blood. And therefore, it increases your blood calcium levels. It's very, very helpful. So here we can see, all right, PTH is going to inhibit resorption of phosphate in the proximal convoluted tubule, so it stays in the tubular fluid here, and it'll work its way through the nephron. Right? Then, right, when we get to calcium, we're going to increase the resorption of calcium in the distal convoluted tubule, so it goes back into the blood. And so as a result, right, we see increased amount of phosphate being excreted in our urine. All right. Now we're going to get to some fun here. We're going to talk about bicarbonates. If you recall back in chapter 2, I know it seems so long ago, bicarbonate is one of our blood buffers, right? That's going to help us to maintain a certain pH in our blood. But also, if we're going to talk about buffering, we have to talk about hydrogen ions. And hydrogen ions will also influence pH levels. Remember this nice comparison, right, between the concentration of hydrogen ions and our pH value, right? The inverse relationship. The higher the hydrogen ion concentration, right, the lower the pH value, meaning that is going to be something that's acidic, and vice versa. The lower the hydrogen ion concentration, the higher the pH value, meaning that substance is more alkaline or basic. So let's talk about our bicarbonate ions. Okay, bicarbonate ions are granted the ability to freely move across our filtration membrane. So they pretty much are filtered right out of the blood, and they will enter into the tubular fluid here. Interesting fact though, right? Any of that bicarbonate that leaves the blood and enters into the tubular fluid is not going to be resorbed. It will not be resorbed. So guess what? We have to pretty much remake, all right, or replace any of those bicarbonate ions that were filtered out of the blood, and we got to put them back into the blood. More about that later on. Okay, more about that later on. So pretty much 80 to 90% of the bicarbonate that was filtered out is going to be reclaimed from the tubular fluid, and I'll show you how that works here in a moment. Right, and then anywhere from 10 to 20%, right, will then be taken up by the thick segment of the ascending limb. Okay, that's the second part of the nephron loop. So keep in mind, this is very important. Any bicarbonate that was filtered out, okay, is not resorbed. We're going to recreate it, or we're going to replace it. And I'll show you how here in a moment. All right, so keep in mind, all right, what I told you about the inverse relationship. Hydrogen ion concentration. The higher the concentration, the lower the pH, the more acidic something is. And then vice versa. Vice versa. All right, so we're going to see, all right, how we are going to regulate the pH of the urine and the pH of our blood. And so we're going to see how that all goes on in our collecting tubules here. So let's start off with acidic blood. So, okay, if something's acidic, that means it has a low pH value. And therefore, if it has a low pH value, then it has a high hydrogen ion. I hate when it does that, by the way. Concentration. High hydrogen ion concentration. So therefore, okay, what we need to do is, remember what I said about the kidneys and what urine is. If our blood is too acidic, that means it has a lot of hydrogen ions floating in it. So we've got to get those hydrogen ions out of the blood and into the urine. Okay, we have to do that. That will increase our pH. Okay, increase our pH. So unfortunately, what we've done is we've offloaded some bicarbonate. We filtered it out. of the blood, and it's going to be floating in the tubular fluid. So we've got to basically make new bicarbonate so we can resorb, all right, that bicarbonate into the blood. So let's start off by looking at the cells here. Our hydrogen ions are going to be excreted into the urine, right, by the type A intercalated cells. Easy to remember, acidic A. So if your blood is too acidic, then the type A intercalated cells are going to, excrete the hydrogen ions into the urine. As a result of that, we will decrease the hydrogen ion concentration. And if we decrease the hydrogen ion concentration, we will increase the pH of the blood. We're making it more basic. Okay, we're making it more basic. And therefore, where did we offload those hydrogen ions? Into your urine. And if we offload them into the urine, they're going to increase the hydrogen ion concentration in the urine. Okay, well, look at what I kind of wrote up here. Okay, that will then decrease the urine pH. Now, the opposite occurs if we're dealing with your blood that's too basic or too alkaline. Easy to remember, the type B intercalated cells, B, think base or basic, right? These cells are going to secrete your bicarbonate, and they're going to resorb hydrogen ions, meaning they will take the hydrogen ions back into the blood, well, as they secrete the bicarbonate into the urine. And as a result of that, this will lower the blood pH, making the blood more acidic. And we are offloading those hydrogen ions, right? And so we'll see into the blood. So there's less in our urine. So that will then cause the urine's pH to increase. I'll show you some pictures here. Right? So here we're going to see, right? Look at this term right here. 80 to 90% of that bicarbonate that came out of the blood gets quote unquote reclaimed in the proximal convoluted tubule. Basically what we're saying is we're going to make you some new bicarbonate. And then 10 to 20 will be reclaimed in the nephron loop. And then of course, depending on what's going on in the body, what the pH of the blood is going to determine, right, what we're going to be doing, right, in our distal convoluted tubule, and collecting duct, right? Whether we're going to be resorbing or secreting our hydrogen ions, or if we're going to be quote unquote, all right, resorbing or secreting the bicarbonate here. All right, so let's zoom in here down at the bottom. Okay, so first step, all right, here's our bicarbonate. It was just, all right, filtered out of the blood, and now it's in the tubular fluid. So it's out here in the lumen. So what will happen is the bicarbonate is going to combine. We've seen this equation before, where bicarbonate combines with hydrogen ions, all right, to make our carbonic acid. Okay, and the enzyme that we use to do that is carbonic anhydrase. And so that carbonic anhydrase then will cause the carbonic acid, right, because of its unstable configuration, okay, it'll quickly fall apart into carbon dioxide and water. Well, those two substances have relative ease moving into these cells here, and that's what they do. Okay, so the carbon dioxide moves into the cell, and then it'll recombine here with with water, okay? And the opposite, we're just going to move this equation backwards now. So carbon dioxide and water, right, they recombine into carbonic acid, and then again, it doesn't stay together long. And so then it then turns into bicarbonate once again, and a hydrogen ion. And the cell quickly offloads that hydrogen ion back into the tubular fluid, whereas the bicarbonate then goes back into the blood. So it can do its buffering thing. So this hydrogen ion then is available for another, okay, bicarbonate ion. So what we'll see is, all right, in the proximal convoluted tubule here, we're going to see a substantial amount, anywhere from 80 to 90% of the bicarbonate that was filtered kind of gets reclaimed, because we did make a new bicarbonate here inside the cell, and then that will get absorbed and dumped back into the blood. Whereas the hydrogen ion is going to stay here in the tubular fluid, making its way towards the distal end of the nephron tubule to be excreted. So we'll eventually pee this hydrogen ion out. All right, so now let's move to the far end here of our nephron, okay, down by the distal convoluted tubule and the collecting ducts in our collecting tubules. Here we'll see, okay, our carbon dioxide entering into the type A. Remember, we talked about the type A. If the blood is too acidic, right, carbon dioxide diffuses across the plasma membrane. It combines with water, turns into carbonic acid. That carbonic acid quickly dissociates. The hydrogen ion, all right, is going to get booted out of the cell, back into the tubular fluid, and then bicarb will go into the blood here, because like I said, the blood has become too acidic. So we need to get this hydrogen, the bicarbonate back into the blood so it can undergo this chemical process in the blood to help buffer the blood. And so again, what we'll see is the hydrogen ion gets booted back into the tubular fluid, and it'll make its way, all right, into the urine. And as it does, it's going to decrease the concentration of hydrogen ions, all right? And as it does that, it'll increase, all right, the pH levels in the blood. But unfortunately, if there's a high amount or high concentration of hydrogen ions in the urine, that will decrease the urine's pH. It'll make it acidic. Okay, so more waste products to get rid of. All right, we've talked about metabolism and how cells, all right, need nutrients for certain processes in the body, and the cells will start to engage in these chemical reactions. Well, as you know, all right, there are going to be waste products generated from metabolism. And one such waste products is going to be, all right, what we call nitrogenous waste. Okay, now nitrogenous waste, right, we're going to see it more, right, in structures obviously that are going to contain nitrogen. An example would be amino acids, okay, and proteins. They have nitrogen, all right, in their molecular makeup there. So when we undergo these metabolic reactions, all right, whatever that waste is going to be, and one of the waste products that we talk about is urea, okay, and that's going to be a waste product that comes from when we are breaking down proteins. Well, that urea is going to contain that nitrogen. And so nitrogen is going to be both resorbed and secreted. And so pretty much we're going to get rid of about half of the urea in our urine. And because of this, right, this helps to generate a concentration gradient. And that will influence the movement, all right, of urea, okay, from the collecting duct and the nephron loop. Uric acid, we talked about nucleic acids, which, and back in chapter 2, right, when we were talking about macromolecules. And so uric acid is going to be a waste product of when we start to break down nucleic acids, and that occurs in the liver. Same thing, uric acid will both be resorbed and secreted. And then we have creatinine. And creatinine, right, this is a great, by the way, substance to use if you want to see, all right, how the kidneys are functioning, because this substance only gets secreted. So if you can give somebody, or something that has creatinine in it and monitor like how much creatinine is cleared by the kidneys, you can have a pretty decent understanding of how much of a substance is being is moving through the kidneys and being excreted by the kidneys. All right, what about drugs? All right, and other bioactive substances? Again, what we'll see is most of the secretion, proximal convoluted tubule, I have to tell you folks, does a lot of stuff. That's the place to be. A lot of resorption occurs there, but at the same time, we'll see quite a bit of secretion going on there. So some drugs, all right, are penicillins, all right, aspirin, for example, will be secreted here in the proximal convoluted tubules. Other metabolic wastes, urobilin, right? We saw that with the breakdown of red blood cells, right? When we were talking about hemoglobin being metabolized, and it goes through the different processes in the liver, and we make it to the kidneys. And this is the part that is going to be, this waste product will be the part that you pee out, right? And then some of the hormones that you see. HCG, human chorionic gonadotropin, that's why pregnancy tests are based on this hormone. So that's why you'll have someone purchase a pregnancy test and pee on a stick, because that hormone is excreted in the urine there. All right, this seems like a good place, right, to take a break and stop for today, because a lot of this stuff here, when we're talking about the concentration gradient and how we establish one, it's a good concept. So I feel like it'll be good for a nice fresh video. All right, folks, I hope you enjoyed today's video and have a great day.